Today we are going to talk about DNA topology, supercoiling and topoisomerases. All these three terms are quite very significant in the case of DNA because DNA topology as well as its supercoiling and topoisomerases play a very important role to maintain the DNA properly stored within the body. At the same time, various functions such as replication, transcription, everything is being controlled by the cooperative action of these three terminologies. Coming to the topology. Topology is a term which depicts how two different parts are being interrelated. Now, in the case of DNA, we know DNA is a double helix. So, the topology refers to how the different strands of the DNA are being interrelated or connected towards each other. That is what we refer by the term topology. And we can see that there is a large amount of DNA which has to be compacted and stored within the cell. So, when DNA occurs as a helix, that is referred to as a coil. That's what is being shown here. Now, in order to store this large amount of DNA within our body or within the cell, correctly speaking, what happens? The DNA has to be compacted again. And for that, what happens? The coils of DNA get twisted one upon each other, and that would result in the formation of supercoils. Now, that is DNA supercoiled. Then, what is the role of topoisomerases? You know that at the time of replication as well as during transcription, some amount of DNA has to be separated from each other. The entwined DNA has to be separated from each other, and the proteins necessary for those physiological conditions have to go and attach to them at the single stranded DNA. But you cannot cut the DNA and make it separated because according to the topology, it has been found to be wound on each other. Now, to remove this or to overcome this, what happens? Certain enzymes comes and helps to unwind the DNA and change its topology so that these proteins of physiological interest can come and bind onto it. And topoisomerases are such enzymes which will help in the unwinding as well as the recoiling of DNA. We will be learning all these three aspects of topology, supercoiling and topoisomerases in this section. Coming to the topology in detail. Now, DNA can occur in different forms, either in a covalently closed, in a relaxed form as we are seeing here in the center. That is, you have the double helix which has been entwined on each other and they are in the relaxed stage. Okay, but when this covalently closed double helix, when it is negatively supercoiled, okay, it can occur in two different forms. When it is wound on each other on itself, when the double helix itself is wound on each other, that will result in the formation of supercoils. It can be either negatively supercoiled or positively supercoiled. Now, what is the difference between a negative supercoil and a positive supercoil? Conventionally speaking, the right-handed helix with having a negative crossovers are considered to be negatively supercoiled. You can look at the figure itself. Now here, this DNA is what? It is a right-handed helix, and that will result in the formation of a negative supercoil. Now coming to this, here it is a left-handed helix. Okay, a left-handed helix, and it will result in the formation of a positive supercoil. Another topological variant which can occur of the DNA is a knotted variant. This is also one which can occur in DNA as well as the tangled or the catenated variant. So, the DNA can occur in different topological forms. Let it be a negatively supercoiled form, a relaxed form, a positively supercoiled, or a tangled form, or a knotted form, depending upon the various physiological states in which it is. So, let's go in detail into what is supercoiling. But we know that always the DNA to be compacted within the cell, to be compactly arranged within the cell, it has to undergo supercoiling. And so we are concentrating right now on supercoiling and the role of various other proteins to overcome this supercoil and various terms related to supercoiling. Now, what is a supercoil? As we told earlier, if DNA is a helical version. So, whenever the rotation of a coil is restrained in some way, it causes the coil to coil on itself. Now, this is a coil of DNA. If this one part of DNA is being expanded and showing here, it is a double helix. So, you have a double helical DNA which is being covalently closed and arranged here. Whenever it gets constrained, okay, what happens? It will get entwined on each other and it will result in the formation of a DNA supercoil. So, the difference of a coil and a supercoil is when a double helix is relaxed, it is referred to as a coil, okay, and when it coils upon itself, that is referred to as supercoiling. Now, a terminology related to supercoiling is linking number. What is linking number? If you have to consider the two strands of DNA separated, okay, the number of times one strand wraps the other is referred to as a linking number. Now, in this case, the linking number is being found to be 1. Whereas in the down strand fragment, okay, in the down portion, you can see that it is linked over here. These two strands are linked at 1, 2, 3, 4, 5 and 6 portions. So, the number of times two strands wraps together or comes in contact with each other is referred to as a linking number. So, in the down figure representing a double helical DNA strand, similar to a double helical strand here, the linking number is being found to be 6. Now, there are different types of supercoiling. These are defined as a change in the number of the base pairs per turn in the double helix. Now, as we know earlier, we spoke that supercoiling is a thing, the coiling of the coil is referred to as supercoiling. But at the same time, this would also result in the change in the number of base pairs per turn in the double helix. That is, when one coil coils over the other, it would also result in the change in the number of base pairs per turn in the double helix. Now, in the case of B form of DNA, there are about 10.5 base pairs per turn of the helix. Okay, and if the supercoiling decreases the number of base pairs per turn, it is said to be negative supercoiling. And if the supercoiling increases the number of base pairs per turn, it is referred to as positive supercoiling. Now here, this is a positively supercoiled one. A positively supercoiled one is always a left-handed helix. Okay, in a left-handed helix, as you see, it's a left-handed helix. In that one, what happens? The number of linking number is also being increased. Okay, when the base number is being increased, okay, the number of base pairs per turn is being increased. The linking number is also being increased. And at the same time, when the number of base pairs per turn is being reduced, it results in the formation of negative supercoiling. And as a result, when negative supercoiling happens, the linking number, that is the linking or the contacts between the two strands, will be found to be reduced. Now, in the picture as such, you cannot depict it, but you can, when the number of the base pairs in per turn is being reduced, that is referred to as a negative supercoil. And conventionally, it is noted that the right-handed helix are being found to be negatively supercoiled. And we know in our physiological state, the B form of DNA is being found to be right-handed and it is negatively supercoiled. Now, coming to the linking number, as we told earlier. Now, the linking number here in this particular figure is being found to be 6. Linking number as such is the number of times that two strands of a closed circular double helix molecules cross each other. And as such, when you say that, it is very simple. And at the same time, this linking number consists of two main factors, the twists and the width. Okay, what is a twist? The twist is a number of times one strand of DNA crosses the other. Of course, when you look into this particular figure, the covalently close DNA, of course, the number of times that it crosses over each other is just the same. So in such a state, what happens, the LK is equivalent, or the linking number is equivalent to the twist. Now what is the width? Okay, and this twist and width together contributes to the linking number. Now what is the width? Width is nothing but the number of sites where the double-stranded DNA crosses over the another double-stranded DNA. Now look here, you are having a double-stranded DNA over here, but this double-stranded DNA is not, the double strand is not crossed over each other, so the width is being found to be 0 in the first figure. But now coming to this particular figure, what happens, you see, the double-stranded DNA is being crossed over at 3 different sites. Okay, so the width is being found to be how much? 3. But here, look at this particular figure, the double-stranded of DNA is being crossed over how many times? Almost 6 times. So the width and the twist, they are quite different. When a single-stranded DNA crosses over another single strand, that is referred to as the twist. When a double-stranded DNA crosses over another double-stranded of DNA, that is referred to as the width. Always, and these two numbers contribute together the linking number. The linking number is always an integer, and it could be 1, 2, 3, 4, etc. like that. And in a covalently close DNA, it is a constant. In totally relaxed DNA, as we noted earlier, the LK is equivalent to the twist.